Japan was built up అవకాశం మనకి జरूरत नहीं है ऐसे Those issues on
So, Ahmed, audible. हाँ यूँ आवाज सुनो अच्छा ठीक है चलो ठीक है
हेलो हेलो अस्सलाम वालेकुम। हेलो
Sir, we'll be starting 5 minutes. Sir, please get it. He is coming.
हेलो अच्छा चला
This guy sign करा. आला दूधी और बुलाएगा दूधी।
अस्सलाम वालेकुम और आदाब। विज्ञान सर्वत्र पूज्यते, यानी विज्ञान की हर जगह पूजा होती है। साइंस के स्कूल मेले का आज तीसरा दिन है। साइंस की तरसील, तरवीज और तौसीक के इस प्रोग्राम में आप तमाम लोगों का दिली खैर मकदम है। इस इजलास की कार्रवाई को शुरू करने के लिए मैं प्रोफेसर फारूक अहमद मसूदी, डीन एकेडमिक अफेयर कश्मीर यूनिवर्सिटी को दावा देता हूं कि वो इस इजलास की सदारत के फराइज अंजाम दें। इस इजलास में 4 मकाले पढ़े जाने हैं, जिनमें सबसे पहला नाम सेंट्रल यूनिवर्सिटी ऑफ कश्मीर के रजिस्ट्रार साहब प्रोफेसर मोहम्मद अफजल जरगर साहब का नाम नामी अव्वलीन है। सबसे पहले मैं मरकजी दानिशगाह के रजिस्ट्रार आली जनाब प्रोफेसर मोहम्मद अफजल जरगर साहब से इल्तिमास करता हूं कि आप अपना मकाला बाउनवान कंट्रीब्यूशन एंड डेवलपमेंट ऑफ बायोलॉजिकल साइंसेज पर पेश फरमाएं। आली जनाब प्रोफेसर मोहम्मद अफजल जरगर साहब।
आप हैंड ओवर कर दीजिए चेयरमैन साहब को, तो देन ही विल मॉडरेट।
ओके, थैंक यू।
शुक्रिया जनाब, आप सबका खासकर मुंतजमीन का तहे दिल से शुक्रिया अदा करता हूं जिन्होंने मुझे यहाँ मदऊ किया, और आप दानिश्वर हजरात जो यहाँ तशरीफ फरमा है, आपसे कुछ सीखने का मुझे मौका फराहम कर रहे हैं। इस निशस्त में जैसे आपने अभी फरमाया कि 4 इसमें मकाले पढ़े जाएंगे, जो मुझे फेरिस्त से मालूम हो रहा है कि 4 मकालों का ताल्लुक हयातियाई साइंस से है। कुछ मकाले हयातियाई साइंस में संगमील या माइलस्टोन्स और कंट्रीब्यूशंस बायोलॉजिकल साइंसेज में, जिस पर प्रोफेसर जरगर साहब रोशनी डालेंगे। इसी तरह से हमारी ज़री यूनिवर्सिटी बराह रास्त उनका ताल्लुक भी हयातियाई साइंस से है, चाहे इल्म नबातात से हो या इल्म हैवानात से हो। दूसरा मकाला इसके बारे में होगा। इसी तरह से फिशरीज, अचीवमेंट्स ऑफ फिशरीज इन कश्मीर, क्योंकि फिशरीज का यहाँ स्पेसिफिक कुछ मखसूस अक़साम फिशरीज के यहाँ पर पाए जाते हैं, खासकर कोल्ड वाटर फिश। वी एक्सपेक्ट इसमें इन चीजों का भी जिक्र होगा। इसी तरह से मेरे साथी, मेरे दोस्त प्रोफेसर बशीर अहमद गनाई, उनका जो मकाला है वो बायोमेडिकल रिसर्च इन इंडिया, जिसकी आजकल मौजूदा दौर में इन चीजों की अहमियत और भी ज्यादा बढ़ गई है कि बायोमेडिकल रिसर्च ज्यादा से ज्यादा तहकीक होना चाहिए। किसी भी साइंस के किसी भी शोबे से हमारा ताल्लुक हो, बहसेत मुहक्किक, बहसेत रिसर्चर, ये चीज बहुत जरूरी है कि उस शोबे की तरसील के लिए या उसको आम फहम तरीके से, आम फहम जुबान में पेश करने के लिए कुछ चीजों की जानकारी लाजमी है। सबसे ज्यादा अहम चीज जो उस तरसील में है वो ये है कि हमारी मादरी जुबान, उस पे दस्तरस होना चाहिए उस मुहक्किक को, उस रिसर्चर को, क्योंकि जाहिर सी बात है, हम सब इस बात से बखूबी वाकिफ हैं कि किसी भी चीज का अगर इजहार करना हो, चाहे मोहब्बत हो, जज्बात हो, खुदा न खास्ता नफरत हो, इन सब का इजहार अगर करना हो तो बहरहाल वो मदर टंग या मादरी जुबान में ही किया जाता है। और खासकर साइंस के, साइंसदानों के, मुहक्किकों के, हम सब पूरी आलमी इंसानियत मरहूनी मिन्नत है। अब अगर उनके काम को आम इंसान तक पहुंचाना है, उसको इजहार करना है, उसको एक्सप्रेस करना है, तो उसके लिए जरूरी है कि हमें मादरी जुबान पे दस्तरस होनी चाहिए। अपनी बात को मुख्तसर करते हुए मैं गुजारिश करूंगा अपने साथी प्रोफेसर मोहम्मद अफजल जरगर साहब को, उनका जो टाइटल है, उनका जो उन्वान है वो है कंट्रीब्यूशन एंड डेवलपमेंट्स ऑफ बायोलॉजिकल साइंसेस। प्रोफेसर जरगर साहब।
थैंक यू चेयरमैन साहब। तो जो है मैं अपनी प्रेजेंटेशन के लिए जो है मुझे इजाजत दे दें, मैं वहां से प्रेजेंटेशन करूंगा। My colleague, worthy chairman, Professor Farooq Ahmad Shah Sahab, Farooq Ahmad Masudi Sahab, my colleague, Professor Bashir Ahmad Ganai Sahab, the Dean, School of Life Sciences, Central University of Kashmir, Professor Yusuf Sahab, the program coordinator, Professor Shahid Rasool Sahab, Professor Zahur Ahmad Gilani Sahab, my other faculty members from the Central University of Kashmir, and above all my dear students, very good morning to all of you. In fact, at the outset, I am privileged and honored to be given a chance to make a presentation on the contributions of the biology and biological sciences across the globe. In fact, I have recategorized as key events in the development of biology and biological sciences, indicating the crux line of my presentation will be same what is there in the earmarked topic. Well, my dear friends, life sciences or biological sciences or biology has evolved over the years from older civilizations to the modern civilizations. The word biology itself basically is comprised of two words: bio means life and logy means science. That is, Ecology means study, that is, it is the study of the life or the life processes. If we look at the biology per se, the word, we see that biology encompasses what already has been referred to is the kingdom of the animals, kingdom of plants. But it is not limited to the biology, it is not limited only to the biology of the kingdom of the animals, kingdom of plants. Then it has integrated or it has expressed itself into n number of sideways studies, sideways sciences, like medical sciences is there, biological sciences is there, biochemical sciences is there, even of late the biotechnological sciences is there. And within these sciences, there have been n number of other sciences which encompasses like immunology, cell biology, embryology, so on and so forth. And each discipline in its own has contributed to the mankind. When we look at it, we see that it is the study of the living life and the living things through observations followed by the rigorously tested and peer-reviewed scientific research method. So science actually is contributions, not only the observational studies, but also it has to be tested. The hypothesis has to be tested. We call that as the science. Okay. Next one. Okay. Now, as I was saying, that the biology encompasses a large bodies of information. But when we look at it, there is a uniformity also, commonality, what we say. And it was, despite the broader scope of the biology, there are some commonalities. Like what we say is in all the biological studies, the cell is the basic unit of life. Genes, which are consisting of the DNA and the RNA, they are the basic units of heredity. Evolution accounts for the unity and diversity seen among the living organisms. And all the organisms, they survive by consuming and transforming energy. And all the organisms maintain a stable internal environment. So, although we say that biology encompasses different components, but yet all these parameters, whatever I was suggesting, these are the unifying components. In fact, these are the components which are common to all, without which we cannot say that it is a life study of the life sciences. The first forms of the life sciences, of the life on the earth, it existed for billions of years before the evolution of the larger organisms. Earlier forms were the microorganisms. The mammals, birds, and the flowers, they originated within the last 200 million years. And modern appearing Homo sapien, human being, what we call as a Homo sapien, have inhabited this plant for the last 200,000 years. So when we look at it, the evolution of the human mankind, or the existence of the mankind, it is a recent history as compared to the history of the life over this Mother Earth. Next. Okay. Now, one of the things, there were ancient philosophers who contributed significantly to the development of this science. And later on in the 15th century, 16th century, there were advances in the microscopy. And because of that microscopical advances, we were in a position to identify more organisms. And we were able to identify those things which were hidden from our naked eyes. And it was in 1838, Sheldon and Sherman promoting the new universal idea of the cell theory. They gave the cell theory that is cell is the basis of life, and cells originate from the pre-existing cell. Lamarck was the first to present a coherent theory of evolution, although it is a Charles Darwin who is considered as the father by giving the natural selection process theory. And then in 1953, the double helical structure of the DNA marked the transition to the era of the molecular genetics, which created more and more advances. So on and so forth. Now, when we look at the modern biology and the advances in the biology, we cannot underscore or undermine the contributions which has been made by the ancient civilization. It will not be, I will not be faithful to name each and every person or score the history of each and every person. But what I will be saying, tracing down, I'll be tracing down this historical perspective through the contributions of certain people. Yesterday also, Professor Yusuf Saab and Professor Rishi Saab, they also develop on this historical perspective. I'll be looking at contributions made by some scientists, made by certain experts, philosophers in the ancient civilizations, and later on what we call as expert scientists. And we'll be figuring out the historical perspective, how this biology was developed. You must have heard about the Aristotle. Aristotle जिसे कहते हैं, 384 to 322 BC. And as I have written, that before Aristotle also, the speculation of their life, the speculation about the origin of the earth and the life, they were already there. But they were all empirical investigations. Aristotle is considered as one of the premier persons as far as the work is considered. When you look at Aristotle's work, diverse works, he has worked not only on biology, he has been a great philosopher. Contributions in mathematics. You look at everything, the contributions of the Aristotle cannot be ignored. When we look at the work of the Aristotle in terms of the biology, we see that he in fact produced documents which contributed to this importance of the zoology as well as botany. The studies on the animals as well as the studies on the plants also. Indicating that he was one of the first pioneers who documented. But somehow, Aristotle's work on the plants was lost during the process. We could not retrieve it, although he gave the major concepts. But he was not in a position to document it. This is one of the important things that you are supposed to know as a biologist, as the scientists, as the scholars. One of the important things that you need to know is that even if there are concepts in your mind, these concepts cannot be taken to, you cannot be accounted for these concepts unless and until you do not document your concepts. I will, during the process of the history, I'll be telling you certain things when you will see the concept had been developed by somebody else, but they were not in a position to document it. That's why the contributions of the discoveries of that particular thing was contributed to the ones, or it was attached to the ones, it was given to the one who has documented things. So next, it was Theophrastus. He was the student of Aristotle, 380 to 286 BC. He is considered as the father of botany. So Aristotle is considered as the father of zoology, and Theophrastus is considered as the father of the botany. He worked on the plants, and there are many treatises available which worked on plants. Then Dioscorides he also contributed to the botany, and he, his documentation was usually copied with the figures. And there were, other scientists like Ptolemy's, Aristotle, Herophilus, and other things. Other persons who were Greek and Roman times, they contributed. It was Galen, actually, here, 129 to 199 AD. He was a surgeon. Imagine surgery being conducted in first 100 years after AD. And he did his surgery on a number of animals, mainly mammals, and his studies on the nervous system were, the studies on the nervous system, they remained in force up to the 19th century. 18th century, sorry. Next. The contributions of the Arabs cannot be ignored in the biological science. Arabs, they did a humongous contribution, and even Byzantine. These scholars contributed humongously as far as the development of the biology and the science is concerned. You must have heard about Avicenna, isn't it? Yes. Now here, what I am doing, I am concentrating more on the medical sciences, isn't it? Aristotle's contributions to the medical sciences. So biology has originated, this medical sciences has originated from the biological sciences only. And Avicenna and Averroes, they contributed significantly to the development of the biology as a science. Then it was Emperor Frederick Ii. Consider, at that time, even the emperors were contributing significantly to the knowledge, embodiment of the knowledge. Emperor Frederick II, 1194 to 1250 AD, wrote a book on falconry, and this was a notable investigation in bird anatomy and behavior. Actually, he collected the material from the ancient civilization, from the ancient books, and then he was able to write the treatise. Then other scientists also contributed to the development of the biology as a science. Next. It was M.D. Lucy, 1275 to 1326, who revived human dissection in Italy on the corpses of the criminals. Actually, the situation was such that, at that time, criminals, it was very difficult to get the bodies. And he worked on the bodies of the criminals and did autopsies on them. And, this developed the further insights. They could find out the organs within the body. They could find out the anatomical situations within the body. And you can say that in 1410, the body of even a Pope, Alexander V, whose sudden death appeared suspicious, was dissected. Why I am referring here, this point is that at that time, when they were working on the humans, our human subjects, there was always controversy. They would not be allowed to do such kind of experimentations on the humans. And it was stopped by the popes, it was stopped by the churches. They would not allow. So in 1410, when the dissection was done on the Italian Pope, this gave some kind of acceptability to such kind of experimentation. And it was, the Pope had died due to some, suspicious circumstances, and he was dissected so that the real cause of the death could be identified. Another person who contributed significantly to the development of the science, biology as a science, was Leonardo Da Vinci. Have you heard about him? 1452 to 1519. Leonardo da Vinci ordinarily is considered as an artist. You must have seen his paintings, world famous paintings, the Mona Lisa. So the Leonardo da Vinci, actually he had no formal education. He had not read in the proper schools. He was a great artist of the age, practiced dissection. He practiced the dissection and then took the paintings, drawings, and painted them with the brushes. What you are seeing on the right side is the anatomical situation, anatomical paintings. Since he was not well read, he could not write his observations. He started to write a book jointly with a young anatomist. His name was M.D. Torre. But M.D. Torre died in harness. While they were writing the book, he died. So this later on, this book was published by B.E. Carpy in 1460 to 1530. And this is considered one of the early illustrated books on medicine. The book included several new discoveries. Vesalius was another scientist who contributed to the development of the medicine. Next. On the right side, if you are looking at it, this is the first botanical. I'm just mixing up the zoology as anatomy, dissections with the botany also, plant studies also. Because I was talking about the botany as well as zoology. It was the first botanical garden was established in Pisa in 1543. On the right side, you are seeing the picture of the botanical garden, which is still there in Italy, in Pisa. And then 1545, Padua and Florence, there was a botanical garden in Padua and Florence. Now, another contribution was made by the Arab authors in 15th century. That is why I'm saying the Islamic Renaissance and Islamic period also gave a humongous contribution to the development of the biology as a science. They contributed immensely. I will tell you this smallpox, which really laid the basis for immunology as a science. It was done through observational studies by Arabs. It reminds me of a thing. The Arabs and Chinese, they were the first to find out the protection against the smallpox. Do you know how? It will look awkward. Smallpox, you must have heard, it was a very dreaded kind of a thing. Wasn't it? What was its aftermath? Ordinarily, it would be deadly. But what was its aftermath? It would leave poke marks on the scars on the faces of the children. Or it will go into the adults also. It will not go on. Particularly at that period of time, it was difficult in the sense that, it was considered as a bad thing for girls, for females. Because if they would be getting the poke marks, it would be very difficult to marry them off. It was a challenge for the parents. So Arabs, what they did, and Chinese, they would actually take up the poke marks, the scabs from the poke marks of the people who had survived the smallpox, and they would rub it into the nose of the normal person, especially their young daughters. And either the smallpox will develop or it would give protection to the person. So they took this chance of causing the disease in the children. But there was an outside chance also that they would be making them protected from the onslaughts of the smallpox. So actually they mastered it and they called it as a variolation. Later on, this variolation became the basis for vaccination. And vaccination आपको आज पता ही है, जैसे अभी हमारे हाल में जो है issues हो गए of the COVID-19, and we got protected ourselves only due to vaccination. So vaccination का basis variolation है, और variolation का basis जो है was an observation by the Arabs and the Chinese. Keep that in the mind. So Arabs and Chinese also contributed to the development of the biology and the science, and by producing a number of the drugs from plants. Now you should keep that in the mind, that the plants, they are a rich reservoir of the biodiversity, and they are a rich reservoir of the drugs or the medicines which are currently in vogue. आजकल आपने देखा होगा कि botany, zoology, biochemistry और बाकी सारे सब्जेक्ट, biological sciences, biochemistry में काफी ज्यादा रिसर्च हो रहा है to find out the active principles from the plants. So concept lies through the works of the Arabs way back in 15th century. William Harvey, he did another contribution, significant contribution in 1578 to 1657. In his momentous publication in 1628 of the destruction of the blood circulation. So he basically gave the venous and arterial system of the blood circulation, and William Harvey is basically assigned with the job. Okay, so as I was saying, these conceptual frameworks with empirical data as well as with the data, they were limited to only those things which were visibly obvious, which were visible to our naked eye. But it was with the instrumentation, development of the instrumentation, particularly with respect to the invention of the microscope by Galileo Galilei, that really paved the way for looking at making an insight in the new world, which were earlier on not visible to the naked eye. So the microscopy actually contributed to the understanding of the biology more, and biological products more, biological science more. Next. So it was Robert Hooke. Most of you must have, because you are from biological sciences, many of you. Robert Hooke was the first one who identified the cells by looking at the cork slices. He looked at the cork slices, your right side में आपको दिख रहा है. He found that honeycomb-like structure. He found that there is a honeycomb-like structure.
सरकार साहब, मेरा क्या कहते हैं फर्ज मनसबी है कि वक्त की कमी की तरफ आपको इशारा करूं.
ठीक है. आज हमारा एक जो है शायद स्पीकर जो है कम है, इसलिए जो है 5-10 मिनट आप मुझे दे सकते. ठीक है. So Robert Hooke contributed. Then there was a contribution of Carl Linnaeus. Carl Linnaeus ने क्या किया? Classification system दे दिया, जो अभी तक हमारे जो है Carl Linnaeus का system जो अभी तक हमारे यहाँ चल रहा है. And then there was a contribution of Louis Pasteur. Louis Pasteur, considered as the father of immunology. Louis Pasteur implemented the vaccination on the ground. यहाँ पे मैं थोड़ी और story आपको बता देता कि how Louis Pasteur did it, and that is an eye opener for you. You should take chances, because chances, the luck favors the one who takes the chance. And Louis Pasteur was one amongst them who basically whose luck favored him, but he was a systematic person. मैं यहाँ पे ये बताना आपको जरूरी चाहूंगा कि एक थोड़ा सा एनेक्डोट देना चाहूंगा कि लुई पास्चर का कंट्रीब्यूशन कैसे वैक्सीनेशन में जो है which was a game changer. To all of you scholars, scientists, I would like to remind you, Louis Pasteur once he was actually working on the fowl cholera. What he was doing, he was taking the blood from the fowl with the cholera and introducing it into the live, uninfected fowls. He would basically introduce it, and the fowls would cause the problem. One day he asked his assistant or researcher, his name was Chamberlain. He asked him to introduce it into the fowls as per the experimentation, and he went off for a long leave of 3 weeks on a long holiday. जब स्टूडेंट ने देखा कि भाई जो है मेरा गाइड जो है वो चला गया, लॉन्ग लीव पे चला गया, तो उसने क्या किया, उसने भी लॉन्ग लीव ली. And but he forgot to introduce this, जो कल्चर उसने उठाया था, जो ब्लड उसको इंट्रोड्यूस करना था, he forgot to introduce it into the healthy form. Now, after 3 weeks, when Pasteur came, along with him, before that, the Chamberlain was the name of the student. He also came. He found the Chamberlain was a very obedient kind of a scholar. He did not. When Pasteur asked him, "What about the experimentation? If I and you would have been his Pasteur student, what would we have done?" Sir, हमने कर दिया. Results वही आ गए. Exactly the same result. लेकिन उसने Pasteur को सीधे बता दिया, "Sir, since you were on the leave, I also took it as a chance that I should also go on a long leave. I kept it on the table. I forgot to introduce." So Pasteur, if I would have been in position of the Pasteur, I would have scolded that person. क्या किया है? आपने जो है पूरी मेहनत कर दी, जाया कर दी. Lekin Pasteur was inquisitive. He told him that, okay, no problem. Introduce it into the fowl. So he introduces his aged culture into the fowl. Right? These fowls did not cause the cholera. There was no cholera in these fowls. He then experimented them. He asked his assistant now to infect them with the live cholera culture. When he introduced it, he found that there was no cholera. This was the basis for the vaccination. What is vaccine? It is heat-killed, attenuated organism, same organism which causes the disease, but it is heat-killed or less infected or less infected. So here is a seed जो मैं कहना चाहूँगा, इसीलिए मैंने थोड़ा इसको elaborate किया कि take chances in your life. Be truthful to your science. And when you are doing the experimentation, be truthful, because your truthfulness might lead to some new discovery. Thank you. Next. Charles Darwin, Gregor Mendel, इनकी contributions आपसे छुपी नहीं है. They have been, Gregor Mendel is the father of the genetics, और Charles Darwin ने evolution theory दी, selection selection theory. Carl Landsteiner did a contribution which is a major contribution. He gave ABO blood group system. ABO blood group system. अभी तक सर्जरी कर रहे थे, लेकिन it would often be fatal. सर्जरी जब होती थी, it would be fatal also, because blood transfusion की अगर जरूरत पड़ती तो blood transfusion तो कर ही नहीं पाते थे, because it will also lead to the fatality. Carl Landsteiner's contribution was significant. Then Alexander Fleming's antibiotics. I would not go into the detail. It is also considered as one of the flagship things that happened. Next. The gel electrophoresis, the HeLa cell, and the contributions of Rosalind Franklin, Watson Crick in the molecular biology known to all of you. Next. The discovery of the DNA polymerase by Arthur Kornberg and his son Thomas Kornberg. DNA polymerase, याद रखिएगा. It is also considered as a game changer in the modern biology. Reverse transcriptase, restriction endonucleases, they also contributed significantly. Next. The PCR, you must have heard about the PCR. PCR improved the gene cloning process, amplified the genes. Smallest amounts of the genes were amplified beyond comprehension. And then the bioluminescence जिसकी वजह से प्रोटीन अंदर जो है फ्लरिश हो जाते हैं, they also contributed significantly to the development of the science. Next, this is the green fluorescence protein. It gives us an indication about what is happening inside the cell. We are able to see the fluorescence, what is happening in vivo. अंदर क्या हो रहा है सेल के अंदर. So it is also considered as one of the important discoveries. Next, gene therapy. You must have heard about कि जो है, there are lots of diseases, problems, which are due to the hereditary problems, which are due to the genes. Gene therapy में there is a concept, and it is being implemented also that genes can be now edited and genes can be transferred, or genes can be replaced. Right? So there are n number of the hereditary problems that can be resolved by this gene therapy. Next, RNA interference. There was another contribution by the RNA interference. Next, which resulted in the CRISPR-Cas technology. CRISPR-Cas technology, you should keep it in mind. This is gene editing. जो gene cutting, reintroducing foreign DNA और किसी भी जीन को हमने जो है अगर कट करना है, टेलरिंग जिसे कहते हैं, ठीक है, reconstituting new genes and modifying genes. This has also resulted in the CRISPR-Cas technology, the latest technology. In 2000, it was basically 2006 probably. Durga and Emmanuel Charpentier, both the ladies, they were involved in the CRISPR-Cas technology. Next, I am not detailing it out because somebody else will be teaching you during the course of he will be discussing it during the course of this program. Animal cloning. Dolly was the first animal which was cloned. On the right side, you are seeing, although she died after 6 years only, then what is so specific about Dolly? We could clone this animal from a somatic cell. What is a somatic cell? Which is a differentiated cell. Right? Which is a somatic cell, which is a differentiated cell. So, memory cells से जो है, memory cells से इन्होंने clone उठाया, ये DNA उठाया, और उससे इन्होंने dolly को जो है बना दिया. This was, when this dolly came into existence, dolly experimentation proved. This created another series of discussion. Ethically, क्या ये ठीक है कि हमने जो है कल humans को जो है clone बना देंगे, human clones? Will it be ethically right? When Dolly died, it also created a number of the controversies. That is that it is not a reliable kind of a technology. Dolly के अलावा जो है there was in Spain they cloned ibex. तो यहाँ right side में दिख रहा है. It had become extinct. So ibex को clone उन्होंने किया from a frozen embryo, and so these two contributions paved a new way for the modern biology. Next, Human Genome Project के बारे में आपको पता ही है कि 1988 it started, 2003 it ended, and by 2003 complete sequence of the human genome was available with us, and since then n number of the genomes have been cloned and n number of the genomes के बारे में हमें information. This is paving the way for futuristic health. Now, जब हमें information है about total DNA, जब हमें information है about the human genome क्या है, we will be able to play with our human genome as per our needs. Next, stem cell research. This stem cell research is also having the futuristic kind of, futuristic. This is the key development, key point. This stem cell, I may not be able to just comprehend more, tell you more because time is running out. Stem cell research में जो है futuristic study. Say for example, somebody is having the diabetes. Where is the problem located in diabetes type 2? इंसुलिन नहीं बनता है. इंसुलिन क्यों नहीं बनता है? सेल्स जो है बॉडी के अंदर जो है पेनक्रियाज में वो काम करना बंद कर देती है. If we are able to make these cells, new cells, and introduce them into the organ, pancreas, they will start to provide the insulin. So this will take us, this will be the new way for, treating these disorders. Next, robotic prosthetic arm. This has been also approved by the Food and Drug Administration in 2014. Now, without connecting it to the brain signals are basically through mechanisms. They are basically able to just change this prosthetic arm and use this prosthetic arm, robotic arm, and the person can now grasp. Person can now take the things in the hand in the robots, and this is being controlled by the neural information. This is another great development. Next, this is my last slide, which is for you to understand certain discoveries which are modern discoveries, but they are having the potential to for futuristic study. For example, you should listen and you should, learn about these from the net or the internet or from the other these platforms. A new organ has been added, which is called dysentery. It has been now officially added as a human body's 79th organ. Mitochondrial DNA spring cleaning could prevent aging. There is a mitochondrial DNA, which we call as the mutated mitochondrial DNA. If we are able to just spring that out, it will increase the aging. 3D printing organs could make organs donation obsolete. 3D printing तो आपने देखा है आजकल जो है जैसे हमने अभी वो लाइफ सेविंग इक्विपमेंट्स बना दिए थ्रू 3D series. Now 3D printing of organs is also possible. Can you look into that? The 3D printing of organs, how is it possible? Scarring could be prevented by converting cells from one form to another form. Wherever there is some surgery, it leaves behind the scars. Now the technology has come wherein there will be no scars. It will be by converting one kind of the cells into the another kind of the cells. Researchers found a new type of the brain cells. Now these brain cells are having a humongous potential. Please look into that and study this. And CAR T cell immunotherapy. This is chimeric antigen receptor. This could be the end of the road for cancer. आजकल CAR therapy is the buzzword. मैं ज़्यादा इस पर डिटेल में नहीं जाऊँगा, but this is the buzzword. और कैंसर थेरेपी के लिए it is being used across the world. 5 अभी तक थेरेपीज जो है वो अप्रूव हो गई है, और if it is perfected, we will be able to, it will be then cancer will be treated as a other mild kind of a disease. But this is for the futuristic study. Thank you very much for your patience.
शुक्रिया अफजल साहब, आपने बहुत ही दिलचस्प और निराले अंदाज में हयातियाई साइंस का एक तारीखी पस मंजर हमारे सामने पेश किया। हमारे जो नेक्स्ट स्पीकर हैं जनाब ज़हूर अहमद डार, जो कि इस वक्त कहीं बिरूनी रियासत हैं, तो इसलिए हम उनसे ऑनलाइन जुड़ेंगे। तो तकनीकी जो यहाँ पर हमारी टीम है, हम उनसे गुजारिश करें कि वो ज्वाइन कर लें। वालेकुम अस्सलाम।
Sir, is it audible?
Absolutely, you are audible.
Sir, thank you very much. I am speaking from Maharana Pratap University in Udaipur, and I represent my director of research, who is busy in sharing this presentation with all the respected faculty and deans and directors. Sir, I'll share my screen. Is it okay now, sir? Sir, is the screen visible?
बिल्कुल visible है जनाब.
Yeah. Visible?
Visible. It is visible.
Okay, then I can go ahead. Most respected Professor Basudi Saab, my teacher, Dr. Bashir Ahmad Ganai, whom I can see in the panel, other dignitaries, Assalamu alaikum, and it's a great pleasure to present the role of SPOS Kashmir in agricultural progress of Kashmir Valley. Sir, our goal, as we all know, is to be the first innovation-led farm university. And for that, what we are doing and what our R&D is, what our teaching is, what our outreach is, I'll try to focus on that. We are intending to be a research and innovation intensive university with a spinning wheel of knowledge, which somehow we aim education with innovation and entrepreneurship, because that we believe is the future to go. And the new educational policy that's being implemented is also aimed at such an endeavor. That's why we have put in a spinning wheel of knowledge. This is the brief of our university. We have 7 faculties. That includes 5 full-fledged faculties, 2 colleges, 12 research stations, length and breadth of Kashmir and Ladakh, 14 Krishi Vigyan Kendras, which cater to the temperate, humid, and cold arid Ladakh. Our endeavors are from the Research Council or the in-house projects to agriculture policies to externally funded projects, which I'll come later on from various funding agencies, advisories and consultancies, release of new varieties, intellectual property and technology management, space schedule, which you all know. We have 69 subject matter divisions, which cater to the mandated activities of agriculture and allied disciplines. This is our brief journey. In the ICR rankings of state agriculture universities, in the last 4 years, we have had a quantum jump. We went from 27 to 6th among SIUs. First time we participated in the Atal ranking and got band one rank in band excellency. Our endeavor is to be in the top 10 general category. And the SIUs, we are aiming for the top 3. Our Scopus H index has gone from 33 to 48, and we intend to push it to 80. This is only Scopus indexed, high impact journal based. We have 5 startups. We aim it to push to 20. 6 patents have already been allotted to the university. 25 is the target in the next 5 years. This is a snapshot of some of the rankings, including the DST Lockheed Martin governed India Innovation Program, in which we got awards and we got grouped into institutes of eminence like IIT Madras. This is a brief of the startups that we have. BARAC DBT funded programs are recently received. Our advantage we want to take home is to leverage this agriculture sector in J&K, and 30% is the contribution of the primary sector as compared to 18 or 16 at the national level. Ours is 30, and the future lies somehow in our fields, which range from horticulture to animal wealth to field crops to fisheries to forestry to sericulture and agriculture engineering for the final process. So this is just a brief idea about the H index and I index and the PhDs awarded. In the last 5 years, our I-10 index has gone from 79 to 110, and our PhDs have gone from 40 in between 51 if we add it. This is the snapshot of what we've been doing. 850 number of Scopus citation index papers in the last 5 years in high impact journals, ranging from scientific reports to PLOS One to Frontiers in Plant Sciences. You name it, we have it all. Viruses. Number of faculty with at least 10 is 60. So the number of SCI papers evens out to 30. 150 PhDs we have awarded in the last 5 years, and 300 are ongoing. That's our strength. Human resource, capacity building, that is our strength. I'll come to that. So this is just a snapshot of the 5 big grants, if you can call it, in the last 5 years. And the biggest has been the National Agriculture Higher Education Program, which our current Vice Chancellor, Professor Nazir Saab, is the PI. We received a grant of 29 crores from the World Bank aided ICR funded program. It's now coming to close in March. And it has been extended without any further financial implications to 2 years because of the COVID. And the total financial outlay which we received was 29 crores. In between, we got some 8 crores, 7 crores, 1.5, 1.1. There are lots of projects which I'll come later on to the strength of the university. And they range from funding agencies like DBT, DST, National Mission, the PPVFRA, UGC, NABARD, International Food Policy Research Institute, DBT Wellcome Trust, DST SERB, in addition to the DBT. Some of the glimpses of the facilities are available. We have tremendous land resource. That's our strength. We have tremendous land resource, 2,000 hectares and more than that for growing crops. We have genomics facilities, metabolomics, drug discovery, short-term and medium-term germ plasm storage we built recently. We have a discovery building for the genomics center available. This is just a snapshot in addition to already existing infrastructure, which is quite huge because we span the length and breadth of the valley as well as Ladakh. Our endeavor starts from here because the presentation is on the role of cost. We are a pioneer in molecular breeding, and this is just a snapshot of the procedures that we follow for gene-QTL discoveries for different crops in wheat, beans, chickpea, and cowpea. In addition, we are doing molecular breeding platforms in rice, maize, and other crops, which I'll come. This is just a snapshot of what we're doing: association mapping, mapping populations, QTL mapping, biparental mapping, you name it, we have it all. This is a recent release variety. This is the first variety in Mushkubudji background released by a normal seed production activity in which we pyramided 3 genes using marker-assisted backcross breeding for rice blast. And on the left, you can see the improved version. And on the right, you have the Mushkubudji devastated by rice blast. Since it's a niche, we already have the award of Genome Saviors from Ministry of Agriculture to our farmers who grow it in Sagam area. It has an issue of it succumbs terribly to rice blast. It's a complete devastation of the crop. And we could introgress 3 genes, PI9, PI54, and PETA into the background of it and improved it. And recently we have released it for the state of J&K. So now it's a release variety, which is an improved MB. It's called Shalimar Rice 6 by the name, which we have improved it for rice blast without any compromise on the aroma and other quality. We mapped a QTL BQ6 of Bakan and another high altitude disease, and the QTL explains substantial amount of variation. We have a caste-based single SNP-based diversity analysis already published with 484 lines from Kashmir, and we are trying to map genes. Previously, we used to map genes for leaf blast, genes like PI9, PI54, but as a global endeavor with International Rice Research Institute, we're trying to map genes for neck blast. And as far as I understand it, neck blast is actually a QTL. It's not a gene only, it's a region which we are trying to map in our background. This is some of the molecular plant pathology work we carry out. And as you can see, remarkable work has been done in terms of apple scab, in terms of anthracnose of beans, in terms of a robust method for screening of transformants, identification of new pathogens using gene-based sequencing, population structures. And for the first time, we discovered the population of North Indian common bean land disease through SNPs, high-end SNP genotyping we have done. And it's a novel and major anthracnose gene which has now been documented and cataloged. The publication stands published in Frontiers in Plant Sciences in 2021. This is a program which we received recently. Our endeavor is that can we have a nutritionally enriched maize hybrid? And we have been successful in releasing a hybrid which is normal in nature. And we are going to go for a conversion program using marker-assisted backcross breeding. And this project has been sanctioned recently to us by DST. And we are going to have an improved version of this hybrid, Shalimar Maize Hybrid 5, which is actually an interinstitutional hybrid between SquashK and Indian Institute of Maize Research. And this hybrid is going to have a large impact owing to its abiotic stress resilience. It stands already recommended by NICRA for its climate smart program across India. It was a part of the project which we did with NICRA, the National Institute for Climate Resilient Agriculture. It got identified in that, and subsequently we released it on 8th of February for the state of J&K. And now that it has been released, we want its improved version for food and nutrition security because maize is a part of the Nutri-Cereals program of Government of India. So normal maize alone not, but we are improving it for lysine and tryptophan. That's how we understand the quality protein maize. And in future, we are going to introduce genes for ProVA. Actually, one of the programs we are going to undertake, in fact, it's already in progress with IRI, Indian Agricultural Research Institute, is to pyramid genes for ProVA, QPM, and vitamin E, 3 quality gene stacking using conventional plus molecular breeding and no transgenics. This is all plant breeding with the aid of molecular markers, so that we don't land in any IP issues or regulatory issues. This is just native host genetic variability, which we are just crossing from one line to another line using marker-assisted backcross breeding. This is some of the achievements of the Animal Genomics Center, which we have. It made the world's first handmade Pashmina goat clone. It first time fully annotated the genome and published it with the ID. Draft genome of Dactylobacter notus published and availability made publicly available. We have the first foot rot vaccine, which is to be deployed only. And we have databases for animal breeds like poultry, pashmina, goat, and sheep. We are trying to have an artificial intelligence machine learning endeavor in the future also. And pipelines have been published using computational biology for genome transcriptome and epigenetic analysis of crop, animal, and human genome. Some of the achievements of the Plant Biotechnology Center, we discovered 6 new wilt-causing fungi. First reports invented Scotch matrix assisted reported gel, which was used to develop soil testing kits. Developed 25,000 molecule library from medicinal plants and DNA barcoded 400 species of liquorite. Some of the technologies which we transferred in 2021, as you can take a glimpse, because this presentation aims what is the role actually. Now the role starts. Previously, I was specific on the achievements of the industry. Now the role is indoor saffron cultivation, molecular diagnostics. Can I request the colleagues to mute, please? Then what's there? This is the commercialization continued in terms of the post-harvest value additions which we are doing. This is transferable technologies in engineering which are ready for commercialization. We have MOUs with so many companies which are into engineering, and this is one of the snapshots of walnut dehuller, apricot fruit harvesting, fruit harvester, tunnels, paddy threshers, graders, vegetable transplanters, cob shellers in maize, which saves a lot of labor. Then we have motor operated apple grader. This is a snapshot of the projects which are going on. We recently submitted 190 projects in the last 2 years. And as of now, we have got 38 as finally sanctioned from various agencies, including the National Bee Board, which goes up to 6 crores. This is the ongoing externally funded project scenario. You can see the AICRP, Professor Masood is well versed with this. From AICRP, All India Coordinated Research Projects. Then there are network projects. Then ICR is supporting us, RKV by NABARD, Science and Technology from J&K, Jalim, Tavi, Flood, DST in Serb and Seed, DBT, National Bee Board, Ayush, Govind Vallabh Himalayan Development, international projects with Rice Research Institute, Maize and European Union, DRDO, MOA, ICMR, ICSSR, IMD, PPVFRA, horticulture, student research in terms of NPDFs and other endeavors. This totals 48 crores, which we have in our corpus from the external funding. 48 crores from 173 projects. This is a graphical representation of the project. DST has been the biggest funding agency for us, followed by DBT. This is a snapshot of the manpower because our endeavor is to generate some amount of employment also. 65 in terms of these projects. This is just the steps involved. We signed MOUs with 13 partners, including SPOS Jammu, BARC. We already have a very strong collaboration with Kashmir University, Integral University, Ayush, NCBS, the Tata Institute of Fundamentals. We are going to geotag all the wildlife resources. College of Temperate Sericulture, Indian Institute of Packaging, EPIDA, and IIIM Jammu. Some of the patents that we have been granted of late, our Director of Research, in whose behalf I am presenting this presentation, has got 2 of them for Pashmina fiber by PCR technique and tabletop paddle operated charka. He's got these patents. Dr. Imtiaz Mortaza has got one for hydrolyzing keratinous waste. Mr. Reto and Malik Mukhtar has got one for estimation of residues. This is the snapshot of the patent applications. Our endeavor is to go to 20. We already have submitted 40 for various scientific technologies that we have generated. This, I believe, the House should take note of this. This is where the real impact of SCOST is in terms of the impact of the varieties that have been released in the field itself. And this data has been generated in collaboration with Department of Agriculture and of course our own Division of Agricultural Economics. And till now, SCOST has released 104 varieties for state and national scenarios or ecologies or niches, whatever you want to call it.
Disconnected. प्लेटफॉर्म खोल रही
इनको आप बताइए आप बता दीजिए सर
Are we audible? Are we audible to the speaker?
Speaker is not audible. I being the farmer, correct. Yes,
Nazir sir, you are not audible. Zohr sir, you are not audible.
Disconnect. Last slide.
Shahid sir?
Just very nice. Very nice.
Sorry, sir, I got disconnected in between. I'll be continuing again.
Please go ahead.
Hello? Sir, is it visible now? Is it visible?
No, your screen is not visible, but you are audible.
Okay, let me share it again. Just a minute. Share. Is it okay now, sir?
Yes, it's all right. It's all right.
Yeah, these varieties which I was naming, Shalimar Rice 4 is equivalent to Jaya in the National Maize Program. The variety has a yield at farmers level. We have harvested more than 9 tons from 1 hectare. It's a huge impact in rice. Same with maize, a little bit in maize, oil seeds, wheat, we rule in these segments. Wheat and oil seeds, rice, we have no competition at all. Similarly with fodder crops. Absolute ruling of in terms of the breeder seed that is given to the Department of Agriculture. Whatever you see in the field, the improvement that you must be seeing in your own residences also. It's all because these varieties have stood the test of time and they have not been knocked down because of any biotic or biotic stress till now. Our endeavor is to make more and more and compete at the national as well as state level. So this is just a glimpse of the additional income that farmers get from these varieties. This is the British Indian which covers the whole of Kashmir. We have covered the whole of the area with this Indian. Some of the R&D issues, I'll leave it for time being. If we get time, then I'll discuss. This is in terms of education, students' participation in the squashed entrance. As you can see, it's steadily increasing. It is 10,000 in 2020, and it has gone up to 11,000 for, this is only for the UG, only for UG. This is student enrollment UG and PG. And as you can see, equal participation. There are 1,800 UG students across faculties. Then there are 700 PG students and 358 doctoral students in our university across faculties. And these are the pass outs, 1,800, 705 and 358. They are expected to complete their PhDs in the next 2 years, but these are all pass outs. These are some of the awards at the national level we have got. The All India Coordinated Srinagar Central Project on Rice with a budget of 1.10 crores, the best TICRP in India. Same with honeybees, post-harvest, NSP. Some of the Krishi Vigyan Kendras who have been rewarded at the national level. I leave it here, inshallah, and it's open for any questions or queries, please.
Thank you very much. Thank you. I think that part will be question answer once all the presenters present their work. उसके बाद हम since he is invited from there.
If there is any question for him, maybe we can take it. question for him?
Any clarification?
Ask him why don't we have other food?
ये कितनी
रास्ता बंद हो जाता है तो क्या
वैरायटी प्रैक्टिस है इस पर
वो शायद डॉक्टर साहब ने कुछ
बताया
Zohar sahab, briefly can
Yes.
you let us know the say number of varieties which have been accepted in different these field crops, be it rice, be it maize, or
I showed it in my slide, but I will repeat it for the forum.
please.
In rice, we have 12. In maize, we have 23. If I can share my slide again, it will be better for the forum. Can I slide it show again, if you want me? Because that will give you the exact number. Just a minute, sir. Just a minute. I'll see. Yeah. Sir, is it audible? My voice?
Yes, you are audible.
Rice, we have 12. Maize, we have 23. Wheat, 3. Pulses, different types of pulses, 16. Fodder oats, 6. Oilseeds, 6. That makes it 66 in field crops and 38 in horticultural crops.
These are the varieties which stand released, but
We stand released at state and national level. Out of them, 12 are national releases.
audience would like to seek the information regarding their acceptance. So the purpose for which those varieties were released, be it disease resistance or yield or better nutritive value. What's the level of acceptance by the growers, by the farmers?
As of now, no variety which we have released, although now we are phasing out the older ones, as you well know, K332, K39, we have not counted them in this slide. We have only counted those varieties which were released after 1982.
So
prior to that, we have not counted. And
Professor Zohar,
we have never faced rejection of any variety,
okay,
as of now. We have never faced rejection either at national level or state level.
so I think that's all because Professor Zohar is from Udaipur. He's from Udaipur. So should we shift another?
Sir, it's very interesting presentation from you, sir. I just want to know something about this. You talk about this Nuri, that is Pashmina code. Is there any results that come to the common people? What is the developments? What about the further research about that? Because Pashmina goat is very expensive, Pashmina shawl here in Kashmir, and the goat is also very less available. The wool is less available here. So that is the Changli something, Changli goat. I think it is not available in Kashmir, maybe available in Ladakh area. So what about the research? Do I know something about that? That research, is it common? It is available to the common people in Kashmir or not?
Yeah, see, the first issue we faced with Nuri was how will we take it to the masses or the people, and how to take care of its purity in terms of the births and other things. For that, we needed a comprehensive genomic coverage to see what are the regions which actually we want to have it. The technique is now perfected, and in fact, last month they had got professionals from Department of Agriculture and trained them to take it forward to the next level. Plus, now that the genome is with us, so we are in safe hands, and I expect that by the time we reach summer or beyond summer, I think September, we will have a major program on outreach of Nuri to the public. Yes, that is already in the pipeline. And we have taken permission from DBT and DST because you need to take permissions because they funded the program regarding the animal welfare and other social responsibilities. That has been already taken care of now. And we are in the process of commercializing. Not only Nuri, will be commercializing the foot rot vaccine also. Foot rot is another big issue in Kashmir. We'll be trying to go commercial with foot rot. Plus, we are in the process of licensing out some of the parental lines of the hybrids that we have developed to Department of Agriculture. In fact, hybrid seed production can be taken up by unemployed youth as a very good endeavor for their livelihood because it has a lot of potential in our area.
Shukriya,
Thank you.
Actually, जुबान के हवाले से एक सवाल
हम कहते हैं वो गिला शिकवा हम में सबसे है जो भी अभी तक स्पीकर्स हैं वो गिला शिकवा सबसे, लेकिन पहले हम जोहर साहब से इजाजत देंगे बिकॉज़ ही इज फ्रॉम आउटसाइड, इसमें कुछ और तहफ़्फ़ुज़ात हैं इसलिए हमें इजाजत देंगे. Thank you, Zohar sir. Thank you very much.
Thank you, sir. It's a pleasure. Thank you very much.
Informative and wonderful presentation by you. Thank you very much.
Thank you, sir. Thank you.
So both these presentations were indeed informative. दानिशवराना और आलिमाना एतबार से, साइंसी एतबार से हमें काफी कुछ सीखने को मिला. इस तरह से हम तवक्कुल करते हैं।
Sir, I have a request. If the house needs, I can mail it to you.
This presentation and
This was
other
my email. If I get a mail ID, I'll mail it to you just now.
kindly send it to organizers and they will send it to others. And that will be
If Dr. Shahid can send his mail ID, I'll send it to you.
Sure, sir. Sure, sir. We'll just
Let him share it on WhatsApp. I'll send it just now.
Thank you.
I'll send it just now.
Thank you. So we are going to listen now another presentation which we expect will be equally informative. लेकिन इस पे एक जुदा किस्म I expect एक अलग किस्म का रंग आपको देखने को मिलेगा. So with these words I request Professor Bashir Ahmed Ganai to make his presentation.
Please.
Yes, sir, now, Satri Marshal. Bigger, those साथियों विगत समन वर्ष में किस मौजूद छे इनके नाइन डॉक्टर सलाम के आदाब को अस्सलाम वालेकुम व रहमतुल्लाहि व बरकातहू
वालेकुम अस्सलाम व रहमतुल्लाहि व बरकातहू
क्योंकि
Michael.
I am giving lectures in different forms, different times with English language, but I am more interested in my mother tongue. That is in Kashmiri. Please get for some time. There is an alam cho Maji Jayhun to Manawan. If I put Che Markaji Jamiat Kashmir Jamia in this Akhwasas. Third Kasar Vidyan is Sadat and Kis Tanazur Asman. Ki Sinus Maji Jayhun Man's Pocher वर्तावत परे तिनिश कोशिश करें यानी पहले हिसाब छे इन हदस्तान इन दशवे यूनिवर्सिटी दशवे जामिया कामयाब कामरान क्योंकि म्योन तकरीर चू तभी तहकीकत पे मबनी तभी तहकीकत पे मेडिकल रिसर्च मेडिसिनल रिसर्च तो बकारे बयान की तहकीक जिन हिंदुस्तान में आजादी ब्रोंट आजादी त्रावन पत करना आम छे मगर अनब्रो मनब कत अक्स कि बचुन ज्यादा बोल अर्लिम के मे छु हुचमुत केसा सहेनिश पन्न बुजुर्गनिश पन्न तीरनिश शेख सदी शिराजी छु वनान दिले खुशबोई दरहमाम रोजी रसीद अल दस्ती महबूबी बदस्तम बिदो गुप्त मुश्किया अंबेरी कि बोए तू दिल आयज़ मस्तम गुप्तार मंदिर नाचीज बुदम वलकिन बंदरे बाबू निशस्तम जमाले हमनशीन दरमन असर कर वलकिन मन हमाम खत्म नवासान तो में ये थोड़ा
नहीं
Sorry, she get over yeah. Or not sorry, I suppose asking for a fake noise coin. Which question? Arsi Zaman hum Sheikh Sadi Shiraz Akbar Bar Shah Akbar Bar Sheikh Sadi Shiraz Akbar Bar Akshar. अगर इच्छुक साथ मसली मत बोलने वान मसला मत बोलने वान दपान अगिदो पुरुष शेख सदी शिराजियन सुन इन प्रश्न बदला दपान पाने यलमस की मासा पुस बुको एजुस मेचे दोबास मेचे तो वंतम सेमस काटी छु युता मुशुक इता अमेरी से काटी आई मुशुक किसकन छी मुशुक क्या के पुराने जमाने में बादशाह करा इस्तेमाल या सावन वने वजूद मस के पता मेचे पोशा थावा मेचे सावन मेचे पता से मेचे सिगन साना वाना करा तो मुशुक बस तो वैसे नहीं इसलिए दिल को थम अरे मोहन दिल को थम से what is to know then तो वैसे नहीं क्या चीज मुझे तो हाँ क्या माने वो जो ना हम बुलंद निश वाई है स्काला से ये सब तो मत ये तो मत ना ऐसे ही मुझे पर्सन को मत ना तो क्या चीज हाथर बच्चे जो है मिक्स ये वो तो मैं खबर क्या जो बच्चे यूं ही निश थोड़ा ये तो कोई मूवीज posture ये साइनस और मुद्दे क्या मनमुत छु क्या छु ना दबान साइनस छु सुअलियम उसालियम ये बेनर निश दूर छु बेनर मतलब वो कुंती कंट्री मंट्री कौन हासल वासल तनिश दूर छु युस आलमी इंसान या तो साइनस छु इल्म छु युसन आलमस गोशरावान छु तो तुमै कोम छु ब्रोआ तकान यमो पनुन सोच तो जहानत अत ब्रो वत बोदु ब्रो पकनव अत लमनच कोशिश करो कि तुमै कोम छु ब्रो तकान तो पकिमिति या पर बकोली एमजीके मनन 2009 मज वनान युसन फिजिक्स गुरबार ओवस द एडवाइजर टू गवर्नमेंट नंबर ऑफ टाइम्स तुचु वनान क्या छु वनान साइंस इल्म छु इंसान के मगजन दिमागो पोषित आत्मन तजस्सुस के एहसास सूज पौध गछित गसन सूज वजूदस यवान तब पर छु दुनियास ये गमस कुदरत के नजारा लबनच तो पतनच कोशिश करा ये इल्म छु तेले फाफलान ये जन अख राह रास या अख सही माहौल आसी तो ये क्या किछु कुनी जोन तहलुल निजाम के ऐसी जाय ठहरो निजाम के बल्कि क्या छि साबित करुन कि बल्कि छि पन कवत महश्र मजाई तो बे महश्र के कदम मजाई हासिल करा ये गो साइंस ये जो एमजीके मैंने पत क्या छु अब पच्ची में क्या लफ्ज वने ये साइंस कितने छि यवान ये कितने छि डेवलप करान तो कितने छि अंदरस मज रोजान ये साइंस पते के कुन जाय के ति पूलित दबान छु बजाबान फारसी क्या वनान अलिमरा दर दिल जले यारे बुवत अलिमरा बर्तन जले मारे बुवत इस अलिम दिलस मज खनित शिलस मज दिलस शिलस मज और इंटर ऑफ द हार्ट खनित छु सोई साइनस छु हमरे यार रोजान दोस्त सिन काम करान यो साइनस पेटी करू यानी सुपरफिशियल वन तो साइनस छु कुछ सरफुल तो आसी वन मगर साइंस का चीवान इल्म का चीवान इल्म व हिकमत जायद अज माने हलाल अशुभ हरकत जायद अज माने हलाल ये इल्म चीवान ये जन पन सिंसियरिटी पन ईमानदारी बरकरार थावा अब छि इल्म सु रोजान मतलब ये कुछ साथ के सब कुछ हिंदुस्तान मज तबीब तहकीकु रुझान आजादी ब्रो तो आजादी प्राप्त पत गोड़ कर बके हम लफ्ज बयान यम आजादी ब्रो हिंदुस्तान मज के तबीब तहकीक तजुर्बा करने आय या क्या कहानी या दलील वजूद मज आय नंबर 8 ये मानने चाहिए कि हिंदुस्तान छु तिम किस फजीलतस मज स्पेशलाइजेशन गोडन्युक दर्जे थावान बिकॉज़ जबान छु ये छु अक इंडोलिस्ट या इंडोलॉजिस्ट इन जर्मनी ओवस जूली 1951 उनसे मना क्या ये हिंदुस्तान छु साइनस मज तिम किस साइनस मज प्रोन साइनस तस्वुर करने यवान तो सबूत के रंची ये मान यवान कि हिंदुस्तान छु किस फजिलस मज बड़ी दर्ज थावान 1894 मज आये पन्ने नयतिश गडनिच आख बहई पाई कॉन्फ्रेंस कलकत्ता मज मनाम आख मज आये नव आबादियाती यानी कॉलोनियल तिबी अमलत तिबी तहकीकिश जान पहचान वक्त मुताबिक तस मुतलिक बड़ी जोश व खरोश सान बहस करनी ये अमे वक्त देसी सुहत आयतन आस आयुर्वेदा तो यूनानी अरूजस पे तहम तोर यूरोपियो नए तर्जु मुतारिफ कर पन्निस नव आबादी लूकन हंद सहूलियत हराम जन हिंदुस्तान मज तजारत के गर्ज आमित पत क्या कोर पत बने ये हुक्मरान पत आई सहूलियत की आम लूकन खतर नेटिव लूकन खतर लूकन रोजान तिंद खतर दस्तियाब था वन मे टेबल बन पहाड़ किराम दिखमित मगर तुम जरूरी कह सहूलियत गई पे इस्तेमाल तो ये आई कैंसर अदार हंदिस सूरत मज दिन ज्यादा पहन तहकीक गई तभी तहकीक जरासीम इलमस पे बैक्टीरियोलॉजी पे गम विलियम लोग लेशमैन तक बुन लेशमैन तलाजार बुनमुत तलाजार क्या लेशमैन विलियम लोग लेशमैन जन स्कॉटिश पैथोलॉजिस्ट तम कर हिंदुस्तान मज ये मुझ फुल शक्ल जिसम यानी बॉडी यानी प्यूम अकिस फूजिस तिल पेटिस यानी पहिस मज जन 1901 मज अमे दाग गुजरो ये पेपर गो 1903 मेयस मज ब्रिटिश जर्नल ऑफ मेडिसिन मज पब्लिश 5 चार्ली डोनियन जन आयरिश मेडिकल ऑफिसर अकिस आम सरकारी अस्पताल मदास मज करान क्यों अकिस बच्चस मज अस्पताल मज डॉक्टर कर आमुत ये पेपर गो पब्लिश 1903 मज जर्नल मज मगर जून किस शुमारस मज If some people are not understanding Kashmiri, then I will shift to English also. There is no problem.
No, sir. This is the first time in which is discussed, sir.
And it is relevant because alam chu, until banawan joy. My Devian door.
Yes. बिल्कुल sir.
उनके इनवाइट दो मनाने खबर चाहिए मैंने बात मैं बसान ऐसे खबर मची आमुद करना इसलिए I am not going to debate my language because I have taken a lot of
Sir, please.
Pakindra Brahmachari. Yum Purushat Tavas Mas, Talazar Khatre, Yuria, Stepamin, Alaji ki Rai, Istimal to. Yuzan wine ki, Winnie, Am Elite Majore, Mani Wan. Which Amaki, Hindustan, Chu, has a wabu, Kala. Asli Mola, and again has a wabu, it's a Padana star. In Zan year, Arnashat Sada. Purushatrovas maz Sheila Daniel Shigar Sapur. Here is a Robert H. Pochen, which Ardasha Toshitas maz has a wabu by the Karamwol Zuria organism. What is Paimanas but all Vladimir Morris Nikai Buluk Hapken? So the Dutch Ardasha Tronatas maz has a vaccine, the tower of vaccine. Tower of vaccine, Tajruba Karne. Here is that. Purushat Shatwas Maz, Pandis, now was spared the Adar Mansub the Alban. After now, half time is due. Panel can't make your Hindi Dash of Ardashat Akshitas Maz, drop sea Waba by the Karne. Use them till Goggle, Dr. Buol, Dr. Kilo, Milawat Kerki, Vajasuit, or the was Gumbu. Purushat Toitras Maz. Those Purushat detail, but actually because shadow of Brom, but Purushat was shy. Ronald Ross, you son, Hindustan Mazal, Yemdan Uti, Sikandar Abati, Calcutta Mas, Tom Gur, Tuchun, malarial zoo, malarial parasite. The parasite was zoo, questions. Actually, make Kanlapus. Good way. So I know Kaujun, and you can Dire Hayati, Dire Hayati life cycle. Because Muhisman, and you can call it when you Purusha Zasman, Nobel Yapte, Shaksya. And again, Nobel Prize, he got Nobel Prize 92 for this simple work. This work, I did not say simple, it was that time simple, but nowadays it is a bit difficult, but that time it was very, very good work. Purusha Soitajan was all Sami Nathan Sidi Dosh, Kala Azaruf, Vyakta, and what the Halu. It is second right much malution. sand fly. So can I much more. Mahir Alia and a Dawan Mahir, Karnal R.M. Chopra, Yimsan Sarpaganda. Up to Vananes, Raufelia, Serpentina. Use that. Up bird, यानी कि अब जड़ी बूटी छे कुछ आसमास कार करनी आसार तो आई मज ओस आप तीनों नए कम चीज या नाच रहे पाएं यूं देन दिल का गिलास करार तो यानी कि वानी
शाबगंधा आशा बुजुर्ग अश्वगंधा
जिसका को संपगंधा after my supplying is a compound which has been actually found which is very useful for anti-hypertensive compound. So Achuvana compound is Achuvana compound is acidified. क्या आप करने जब आप ताज खाओगे दिल डाइट खाने असर हो सकता हूँ चीज़ में MOT आएगा कि जो आप कहने तो क्रिएटर माहिर तो मास्टर इमोकर इक्वटे कलकत्ता तो कराची में कुरुक्षेत्रवास में मलेरिया मुताल BC रॉय यम जान मोशिर जमान तो जड़ी बूटे हैं ताकि कि बानी कार छे वो बड़े देसी बंगाल की इंडिया मुतारिफ करनो इन मजा अजुक जदीद नए तो छठा दवा तैयार करनी चाहिए क्या ये पुरुषत पांचरे पुरुषत अगस्तान दौरान रेमेटिक ट्यूब तब ठीक है तो रेमेटिक दिन स्टार्टेड हो सबूत बाइकल आदरी प्रावण पते ये तभी तहकीक पुरुषत पंचायत के पुरुषत शेख के दहलिस माज वुच डॉक्टर एस पेंटलन वाइसरल मजुमलाई J receptor. So these are some very protein compounds found on the membranes of some say organs, membranes of some say cells which are helpful in getting the again receiving the signal from some different molecules and then doing their mechanism through signal transmission action. आपको वहाँ simple as mass पुरुषत पंचायत की दहली में उचो क्या उचो ये visceral receptors the J receptors को बताया पुरुषत पंचायत में पुरुषत शेड की बेस दहली में उच डॉक्टर रामालिंगा स्वामीन आयोडीनिश कमी गुजारितिक उजुरात बेजुल जनाना मास शिष्टनिश कमी तो ये पेपर को इमो पब्लिश इंटरनेशनल पैथोलॉजी में किस विदालस में पुरुषत पंचायत में SNDN को हेल्थ की जहर यानी एक टॉक्सिन आप पालगा एंड हेल्थ की जहर शनाख्त पुरुषत शेटा में which डॉक्टर बीके आंदन फीडिंग टू स्कैटी मरकज शनाख्त एक बे उचुक इनमें लोग फिजियोलॉजी स्कैटी means कि मतलब how much you are seeing, how much you are feeding the fullness of your stomach, and second thing is what is called as the feeding centers, which are giving you just providing you information where from, whether I am eating enough food or not, and whether I am feeling scatty or not. So Timothy Muchium, Purushata Shetterman. Purusha Shet, Purusha Sadaki Daiman, this doctor or why doctor Shetterman? Ajay Rai, Malhotra, as Pentel, you was Thailand giant pen Gasmus Suchi, Shushik Varam, and Zanka is it. Shushik Varam Kasan, edema of lungs, and Zanka is it. Pusha share, Pusha Sakti, Dairy Mas Go, Warrior Doctor, Atmado Darba Dostur, Virama Murti, Naushira Vadia, Ian Tendan, Tondo Sakyo, Neural Silich, Zankari. ये अभी तभी तवसियात इलाज डॉक्टर बीके बचासन को एंजाइमेटिक गुजरात मुजाहिर यानी एंजाइमेटिक डिसऑर्डर्स पुरुष शेड के पुरुष सत्य दहले मजबूर शरीर में चार्यन कोशिका यानी कोशिका means some sort of disease where we are going to help to treat and after nutritional deficiencies तो कोशिका बीमारियों में जिगर चुना क्रहन मत जिगर के कोशिका यानी we are taking in Urdu कलीजा but in Kashmir we get the hemalti या we get even जिगरी पैथोलॉजी बचन हिंदी जिगर दाई के सिरोसिस बनाची तभी पैथोलॉजी दिमाग में वरम यानी दिमाग के एडिमेट पैथोलॉजी भोपाल गैस और जहर उत्पाद यानी वजह के बनूं यानी कैसे बनियो तो क्या जो लीक क्या जो बुद्रा आजे पुरुषत शेटमस को CGS आयरन जब दोपन हिंदुस्तान में गए BCG काफी पुरुष 70 मास डॉक्टर जैकब जानन तो डॉक्टर बलायन वुच यमो को टेस्ट जबान पोलियो वैक्सीन खुराकु नाकामी निजार तो दौरान गो एड्स HIV एड्स वगैरह बेहतरी ते नमूदार तो गॉडियो थैलेसीमिया मरीज जो जन गैर बाहरी रूम को सोस हिंदुस्तान गॉडियो पेशेंट यानी कि गरीब पता आई वारिया हिस्सा पे थैलेसीमिया मरीज लबन यम अति निगरानी मरकज निगरानी मरकज 1970 मन मम्मी तो दिल्ली में तो बाकी जायन तैयार कर वालदेन हिंद तो स्वदेशी फेडरेशन हिंद अखवास सूल सठा राहत जेनेटिक तगीरात यानी कि जेनेटिक म्यूटेशन इतला आव मैसर करने तो थैलेसीमिक डाटा बेस आई कायम करने जेनेटिक तरीकों सूट आव दायदुक दरियाबत करने जरूरत वो उसने तो थैलेसीमिया निगरानी आई 5 साल मनसूबस बहिस 5 साल मनसूबस मध्य दर्ज करने वन बेके काने यम हिंदुस्तान सठा मशहूर चीमन कामे मन गो कोविड वैक्सीन मुहिम हिंदुस्तान मन
उम्मीद छी के वक्त की पाबंदी थोड़ी तो
मतलब कुछ आगे कर के बता दो बट टाइम चीन होना कितना चलो अपने ऑलरेडी कुछ टाइम दिया ना बहुत सारा टाइम देते हैं ना ये प्रेसिडेंट का भी है ना
इससे बेहतर
कोविड वैक्सीन खुराक आव 10 जनवरी 2021 मार्च दिन तो 10 फरवरी 2022 तान आव 1 अरब तो 63 अरब यानी कि 1.63 अरब खुराक कुम पाँच दिन ये इतला यानी कि वजारत सेहत और खानदानी बहबूद हिंदुस्तान मान यम 15 वोर योर यम एलिजिबल तकरीबन 95% आबादी को कम से कम 60 हासिल तो 75% आबादी हो 75% आबादी हो वो तकरीबन 10 यानी कि तमाम उसमें वैक्सीन डोज दिवाने हुए तो मेरे हासिल हिंदुस्तान को बड़े ऑक्सफोर्ड एस्ट्राजेनेका वैक्सीन यूजन अदारे सीरम इंस्टीट्यूट इंडिया के लास्ट तार बनाना आव तो कोविशील्ड के बनो डाक वैक्सीन इस भारत बायोटेक बनो मतलब वैक्सीन है ट्रायल यानी कि महलवारी जांच चला तो मन पर थोड़ा सा तो मन पर कैसा तो ये थोड़ा सा के फार्मर यानी कि पानी सतह टेबल्स की शक्ल में वैक्सीन को क्या को हमेशा खबर में तो बोल रही जरूरत जो मानून क्या मगर कोई अगर एक कोशिश बोझो याद थयो कैसे पर रोज चतस क्या इतना बीच मतलब मार जाओ चतस रोजान बाबी याद रोजान चतस से याद मुझे मारिया है तो मतलब वो जिसने कोई अगर इन्होंने अलीमरा बात ने यारे बुआ इनको पता याद था उनका बड़ा मुश्किल है अलीमरा बात ने मारे बुआ पता मुश्किल पता नहीं तो मारे बुआ इमा मर्दों हिम तकरीबन 80 करोड़ 63 लाख 3,770 जनान हिम युवा कोविड को 30 करोड़ 99 लाख 6,763 बाकी 4 लाख 5,300 अरब अहमद को सारे को ज्यादा कोविशील्ड को इस्तेमाल 1 अरब 46 करोड़ 33 लाख 63,802 वैक्सीन 72 करोड़ 99 लाख 7,750 तो बाबू स्पुतनिक 5 तो 12 लाख 3,957 को तो वही में ज्यादा गसन क्या आखिर तो आकार तो 3 लाख में 14 लाख करना है हम पहले करे भाई मन बात थोड़ा नतीजा माटी थोड़ा टाइम अगर 5 मिनट ज्यादा से अगर क्वेश्चन में करो सारा तब मुश्किल आसान अंग्रेजी में कहना अगर यकीन बोझो बात खुदा करो मुझे मैं अपना जुदो खत्म आप रात दो नए तरीके हयातियाती तो बेयती भी ताकि सालमती हयातिया दैट मीन्स मार्केट बयानी सालमती हयातियात यह मास नए नवेटिक साइंसी तजुर्बात मिसाल जीनोमिक्स प्रोटोमिक्स ट्रांसक्रिप्टोमिक्स तो बेई के ओमिक्स आज मेटाबॉलोमिक्स तो दो अलग उसमें तजुर्बात शामिल छे हाली अनकी पेश रखने सूच सब लुकन मज ये कहता पे भरोसा करना पे कुलजन हिश पद क्या ये अलिम कित पट हेकि कुनै जोन तभी तक की शक्ल अख्तियार करे तो बेई पुराने जमाने क्लासिकल यानी कि पुराने इलाज तरीके कि तुम है कि ये तुम इज्जत यानी कि तुम ये कोई काम करे क्या करा क्या मतलब यू द पोटेंशियल सो मच पोटेंशियल इज देयर आजकल छे निने जंग बदल जीवन तो अनके मसनूआत है कत करने यवान तेरे वनान अगर ऐसा निने सांग जटो पत्ती गसी जनरेट वगैरह वगैरह आज जीन रावण जीन जरी सोरी कुछ यवान मनाना आज छे मानकुल एंटीबॉडी तो प्राणजी गगरा हिस कत वंकर ये बड़ी दर्जे की तहकीक करने खतरे छठा मौजूद छे तो इन सूची तहकीक करने यवान यूजर बड़ी पैमाने पर नहीं कबूल करान छे बड़े थोड़ा हवान कैंसर दूर हो जाए वन मुस्तकबल यानी इन वो जमाना में क्या हटो इस के ये ज्यादा जरूरी बोझ वो मैंने क्यों मौजूद बिल्कुल बात के बारे मौजूद अगर ये खुश आयंद कत मुल्कस मन तभी तहकीक नई सारी दिलचस्पी लबान मगर ये वन थोड़ा असली पानी कह शोबन मन यह बोड़ मुल्क आने की वजह हवस थावान खासकर बदलून समाजी तो इक्तिसादी हिंदिस तनाजुरस मन यह खुशकिस्मती की मुल्क मौजूद हालत इस असल के अमे किस्म कन तहकीकन मज बड़िस पैमानस मज फरोग दिन कोशिश कर मुल्कन मन बड़ तजुर्बकार तभी अमल मौजूद तो सह हाल कन वरियन मज